others to introduce me as well as introduce some of the aspect of my talk. So I would like to start sharing some of the slides and then talk with you. My talk would not be too long, but it is really very important topic chosen by MSI in this evening. So after my talk, I hope I shall have enough opportunity to discuss with Indian and other global researchers who have been working on microbiology to find some new opportunities for international collaboration. So I am going to share some of the slides. I hope you can see the slide, right?
Yes, sir.
Okay, thank you so much. I welcome again to all of you to my talk on wheat blast, a threat to global food security. And I welcome from my Institute of Biotechnology and Genetic Engineering of Bangabandhu Agricultural University to this evening. My talk consists of a brief note on research in microbiology in Bangladesh, maybe around 4 or 5 minutes, and then I shall tell the story, a bad story of wheat blast, a serial killer on the run, which we experienced in 2016, and it is now settled in Bangladesh. I shall tell our story of genomics, open data sharing, and open science for rapid determination of the origin of the first outbreak of wheat blast in Bangladesh, and our genomics, genome editing, and international collaboration for the mitigation, for the tackling of this wheat blast in the changing climate, and also try to discuss our philosophy of open science and international collaboration for facing any plant health emergencies based on our experience in wheat blast. In Bangladesh, microbiologists are very active and they are working in various areas. Major foci of microbiology research here, microbial diseases in humans, and especially a cholera pathogen. Bangladesh is very famous on doing research in this deadly human pathogen. Disease pathogens in livestock and poultry. This is another very important area of our research. Plant pathogens such as blast fungus that I am going to tell the story, our current updates. And we have been also doing research on beneficial microorganisms such as probiotics of plants, probiotics of animals, and others. One of the leading institute in our country is the International for Diarrheal Disease Bangladesh, which is called ICDDRB. And this research is premier research institute, a frontier research institute in doing research in diarrheal diseases. They have various focuses like epidemiology, clinical investigation of the diseases, evolution of the cholera pathogen, and other antimicrobial resistance, vaccine development. And we have some world-leading researchers in the ICDDRB like Dr. Firdousi Kadri, Dr. Farooq. They published dozens of papers in Nature, Science, and they are leading in this subject. Other research, microbiological research, have been carried out by different public universities like Dhaka University. They are mostly focusing on different human and animal pathogens and different aspects from genomics to the phenomics, as well as molecular biological research are carried out in different universities. Research in agricultural universities and research institutes are prominent, and they are mainly focusing plant diseases as well as animal diseases. And also one of the area is probiotic bacteria. Since the independence of Bangladesh in 1971, Bangladesh did outstanding work in the field of agriculture, especially the translational research. We increased our food production since last 50 years. This year we are celebrating 50 years of the independence, and we increased our food production 3 to 4-fold by the application of research and research-based knowledge and technologies in the farmers field. It is an amazing success, and Bangladesh is nearly self-sufficient in cereal and other food crops. Our scientists during this COVID-19 pandemic, they are doing different types of research, mostly genomics as well as bioinformatic analysis of the severe acute respiratory syndrome coronavirus 2. And you can find hundreds of papers published by our microbiologists, especially the young microbiologists. I congratulate all of them. Now let me start my talk today. I am going to talk on wheat blast, which is a deadly fungal disease of plant, and it is called the killer of wheat. And before I talk on wheat blast, I would like to share one slide on the emergence of fungal diseases in the last 50 years. You can see the emergence of fungal diseases are increasing very fast. This paper published in Nature, you can see how faster the emergence of fungal diseases are increasing. Although we have modern agricultural practices, diseases of the major food crops up to 15% pre-harvest yield loss, and saving of this much of a huge crop yield could help us to feed millions of people in this earth. Now I'm entering into the main topic, that is wheat blast. That is one of the emerging fungal disease which first spotted in Bangladesh in 2016. And already you heard from the introduction session that 15,000 hectares of our wheat crops suddenly devastated by that disease, and yield loss was up to 100%. We are not at all familiar to this disease. This is why agriculture ministry decided to burn the crop to clear the inoculum in the field. And here you can see the 8 districts. They are our southward, very close to Indian border, and another 2 districts in the south of Bangladesh. And till now, after 2016, the disease spread 20 districts out of 64, but the severity of the disease was not too much. When I first saw the news in a Facebook post by Sufyan Kamaun, one of the greatest biologists and microbiologist. He has been working in the Sainsbury laboratory, my good friend, because we have been working on omicity genomics and genetics since long. He shared this news report published in our national the Daily Star, wheat blast threatens yield, and he expressed his concern. And this was a first notice I received about the wheat blast. And what is the wheat blast at that time? I didn't know anything about it. And then immediately I communicated with our agricultural department, extension department, to get the news. Here you can see the symptoms of fearsome wheat blast in Meherpur, the district which was severely affected by the 2016 wheat blast disease. The CMIT and other organizations, they are visiting 100% yield loss. All the spikes became white and there were no grain. And I received lots of information from our field agricultural extension officers. And I also received this image. Here you see burning of officially burning of the wheat crop to clearing the pathogen in by the decision of the Ministry of Agriculture. Burning also created very big panic. So it was a big anxiety for our country, because Bangladesh, you know, very small country, but our population is 170 million. So food security is always a big challenge. After having such news, we tried to get the information about wheat blast. Wheat blast is a fungal disease caused by a filamentous ascomycetic fungus, Magnaporte oryzae, which is also known as Pyricolaria oryzae, and it emerged for the first time in Paraná state of Brazil in 1985, and then it spread in neighboring countries like Bolivia, Argentina, and Paraguay. And it is a big threat to, savanna region. 3 million hectares of wheat farmers, they are always scared about this fungal disease, because in ambient condition, if weather favors, yield loss can be 100%. This image I received from Professor Andreas von Tiedemann, who worked in Brazil on wheat blast. He was my host when I was at University of Göttingen as an Alexander von Humboldt fellow. Here you can see, as long as you can see, 100% spikes of the wheat became bleached and no grain there. So horrible disease, and you cannot compare with any other disease. Wheat blast, obviously a deadly baffling fungal foe, and it spread by seed, crop residue, and also several kilometers through the wind. And already it's the conidia is like 3-celled conidia. This disease is caused by Magnaporte oryzae, a fungus I told you. Magnaporte oryzae can infect 50 species of the Gramineae family, or Poaceae, or grasses. Among them, here you can see this is the lineage of Magnaporte oryzae. They have the different lineages, for example, Lolium lineage, or Triticum lineage, or Oryza lineage. Oryza lineage can infect the rice. Triticum lineage infect the wheat. Wheat pathotype never infect rice. Rice pathotype never infect wheat. And they have lots of other lineages. This and it can easily mutate and difficult to control by the fungicide. And once, as it is a wheat blast is a disease, a head disease, once you see the symptom, the vascular bundle is blocked by the fungus at the neck of the spike, and if you kill the fungus, you cannot save the crop. This is why it is a very big problem. And you know that wheat as a food crop, very, very important, because it offers 20% calories and 25% protein consumed by humans worldwide. In Bangladesh, it is the second important cereal crops after rice. Present demand of wheat in Bangladesh is 7 million metric ton, and it is increasing due to the dietary changes. But due to wheat blast, Bangladesh ranked fifth in wheat importing country because our wheat production dramatically decreased in the districts where wheat blast devastated. Here you can see the data of wheat production worldwide. China is #1 wheat producing country, and India is the second highest wheat producing country in the world. And then Russia, USA, France, Canada, Germany, Pakistan, Ukraine, and Australia. So in Asia, China, India, Pakistan, and other countries producing significant amount of wheat, nearly 40%, more than 40% of global wheat. As the pathogen is seed-borne as well as wind-borne, this is why it is a big threat, and there is a high chance to spread of this disease in these major wheat growing countries. This is why it is a big worry for the food security. How I started and engaged on this, wheat blast research. When I saw the Sufyan's post, I had conversation through Messenger, Twitter, and Facebook with Sufyan, and we decided to figure out about the molecular identity of the pathogen. There were and we immediately formed a group with Sufyan, Daniel, Diane from John Innes Center, and Daniel Kroll from ETH, and we formed a group to do the research. What kind of research? We had a hypothesis that wheat blast in Bangladesh may be caused by a native blast fungus from rice or other grasses through host germ. That means through mutation. In case of wheat blast, such evidence are there. Or wheat blast in Bangladesh may be imported from South America through seeds, because it is available there since 1985, and it is a seed-borne disease. And what approach we followed? We followed the field pathogenomic approach for determination of the genetic identity and the origin of wheat blast, because field pathogenomics is the sophisticated technique. By using that technique, you can cover a large population of the pathogen without isolating the fungus. You can determine the genetic identity very precisely. So field pathogenomics, it is obviously a new method to analyze fungal disease from the field samples and pinpoint the exact genotype in a faster way. And we collected the sample for the field pathogenomics. We collected the samples like this way from the field and on the RNA later solution, because our target is to extract the RNA. And then we sequenced the RNA by using next generation sequencer, and then we assembled the short reads by using Trinity software. And our target was to compare the sequences with the known MagnaPorte sequences available in the gene bank, but the problem were there, big problem. We found only one sequence available in the NCBI gene bank on wheat blast fungus, BR32. All other sequences done by 3 decades by the American and European researchers, they locked in their website, and it was out of our reach. This is why we changed our strategy. What we did, we issued a press release. Scientists issue rallying cry for wheat blast research. We released all of our data through the open wheat blast and made a call to the global scientific community to join there. Here you can see 12 samples. The sequence data as they were released from the sequencer. So anyone from anywhere can download the data and compare the sequence data with the data available in their laboratory. So it worked, and every week we are giving the updates. And interestingly, our call, that is press release, was duly published by Nature, made 2 pages news, as well as the Science and many other news media. They covered our call, and immediately scientists from Europe, America, South America, they responded and they downloaded our sequence data and compared with their data, and finally we identified the enemy of the wheat. And it was a big news in our #1 newspaper, Prathomalu, that is enemy of wheat detected. And we detected the here you can see the data. We detected the fungus, as here you can see the real data. We compared our data with different blast pathotypes, Magna porteurii, and finally it was found that our isolates are very closely related to the Brazilian isolate. Unfortunately, Bangladesh imported a large amount of wheat just 2 years before the outbreak of the fungus from the Brazil. And you see pathogen, all the isolates that we sequenced, we found they are clonal and they are very close to the South American wheat blast pathotype, that is a Magna porteurii triticum pathotype. And we started our data with BR32, but due to open data sharing and open science approaches, finally scientists from 4 continents, 31 researchers joins, and here you can see a big list of blast fungal isolates. They were matched with our, they were included in our study. And we also isolated the fungus in the laboratory, and we found that it can infect the wheat but not infect the rice. But there are some other grasses, artificial inoculation show the susceptibility. Immediately, within 6 weeks, we published this paper. Emergence of wheat blast in Bangladesh was caused by a South American lineage of Magna porteurii. We had no project, no connection. You see, using global digital media, social network, we engaged 31 researchers from 4 continents, and immediately we shared our findings through the bioarchive portal managed by Cold Spring Harbor Laboratory. And within 5 months, we published our paper in BMC Biology, a flagship journal, as a peer-reviewed journal of BMC. And this paper already been cited 200 times, I checked today. So this discovery helped us to suggest our farmers what to do, because in South America, this disease has been there since 1985. So they adopted lots of agronomic and other management strategies, although there are no resistant varieties. We immediately released a fact sheet to our farmers so that next year they can face, they can take enough care. Here, engagement of scientists from the contributing scientists of our paper. And here we analyzed the weather data, because why 8 districts were devastated but other districts remain unaffected. We found that our wheat growing season is winter. Winter is free from the rain, but at that year in February, suddenly unusual rainfall accelerated the devastating outbreak of the disease. And here are the symptoms of the disease in 2017, 2018, 2019, and 2020. 20, that means this disease settled well in Bangladesh, although the damage was not as much as it was in 2016. Now 20 districts of our country are contaminated by the wheat blast fungus. And our wheat production also significantly decreased, as well as import is increased. Wheat blast next year in 2017, it was in the Hindustan Times reported that in the neighbor of Bangladesh in West Bengal, suspected wheat blast was reported. And wheat blast also arrives in Zambia in 2018, and paper has been published in PLOS One in 2020. That means it is now in Africa, now in Asia, now in South America, and there is a high chance of spread of this disease. This is the paper published on the wheat blast news in Zambia. And the challenge after discovery, the big challenge was to have a diagnostic protocol for monitoring and surveillance of the wheat blast fungus, and there were no precise diagnostic tool in 2016. But 2017, American researcher in plant disease, they published a paper, and they claimed that MOT3 primer is very specific for the wheat blast fungus. So we thought it is very good, and we can use this one not only in the field, even in the quarantine it can be used. But when we tested MOT3 primer in our laboratory, as well as Sufyan's lab in UK and in the Switzerland, we found that both rice blast as well as wheat blast, both are amplified by in the PCR. That means it is not so precise. So we need a precise and specific and convenient diagnostic tool to detect and diagnose this pathogen. So we published a paper, cautionary notes on the use of MOT3 diagnostic assay for Magna Porte oryzae wheat and rice blast isolates in phytopathology. And we decided to develop and find the more precise and specific primers. Here, in collaboration with Professor Guliang Wang of Ohio State University, as well as some scientists in Chinese Academy of Science and USDA, we by mining the genomes of Magna Porte oryzae, we found two segment. One is MOT6089 encodes a putative acid trehalose protein, and another MOT6099 encodes DUF341 family protein. They are very specific and they are unique in the MOT, that is Magna Porte oryzae titicum genome. And we published this paper in Engineering Journal in 2020. And when we used in the PCR, we found it is very specific to detect the fungus in the plant, as well as in the grain, as well as the grasses, and we validated it in our laboratory. Then PCR is not convenient for the farmers as well as field workers, or even in the quarantine. This is why CRISPR-Cas is a revolutionary technique developed by Emmanuel Charpentier and Jennifer Doudna, and they got Nobel Prize in Chemistry 2020. And we found that one paper from the Harvard University, they use Cas12a enzyme and guide RNA to detect the human pathogen. Then we decided to use this smart technology to develop a strip-like, a pregnancy strip-like method to diagnose this pathogen. And finally, we developed it. Here you can see this strip. If you put in a buffer solution with crushed plant samples, you can find the single band if there is wheat blast, because Cas enzyme can digest the targeted the segment in the MOT fungus, as we described earlier. So this is a very amazing technique, and within 30 minutes you can detect the wheat blast in the seed lot or in the plant. And I believe it would be worldwide use, especially wheat is #1 export import crop. This is why in quarantine as well as field and research lab it will be used. This is why we are now going to sign an agreement with a big industry who will market this PCRD strip in a cheaper price for the mass use worldwide. So our next target was to develop a blast resistant variety. Wheat is a hexaploid, and genetic engineer always avoid the wheat for genetic engineering. And over the years of research, scientists discovered some of the genes responsible for the blast resistance, but most of them, they are not stable at higher temperature. Bangladesh, a tropical country. And once a chromosomal segment 2NS translocation, which was discovered from Azilov's ventricosa tetraploid wheat and RMG8 and RMG-GR119. These three, genetic makeup are considered the influential for development of the blast resistance. But 2NS translocation practically in Bolivia and Brazil initially showed high resistance, but gradually it was eroded. Some of the blast isolate eroded it. And this is why it is not so interesting for us. We considered RMG GR119 because Professor Tosha of Kobe University, he developed from an Albanian origin, discovered this gene, GR119 genotype, and he gave us this genotype. And Bangladesh recently, in collaboration of the CIMIT, released a variety, Barigom 33, which contains 2NS translocation. It is 70 to 75% tolerant to wheat blast, not totally, resistant. This is why how to develop a blast resistant variety is a target. One way is consider RMG GR119 gene discovered by Professor Tosha. But gene transfer in transgenic is another big problem, because golden rice, which was discovered in 20 years before, till it is not in the field. So people have some confusion as well as controversy on GMO. This is why we decided to develop novel blast resistant wheat variety by using CRISPR-Cas technology, genome editing. Genome editing of what? Because several scientists, especially Sufyan Kamaun, he is one of the champion in application of CRISPR-Cas technology. He applied just after the discovery of Jennifer and Emmanuel gene editing technology. He applied in the plant and successfully edited the plant gene. And we decided to edit the S gene, susceptibility gene, that is negative regulator of the gene. And we got a good fund from the British Biotechnology Research Council. Nick Talbot from the as well as Sufyan Kamaun, they visited our country and joined with us, and we edited 10 S genes by using CRISPR. And then we tested them in the laboratory. Some of them showed moderate to good resistance, but as wheat blast is a head disease, this year we shall test them in the spike, whether they are resistant or not. And another scientist from National Research Council of Canada, Pankaj Bhaumik, he developed a very nice protocol for wheat transformation mutagenesis by using CRISPR-Cas, and he also edited by using our construct the Canadian some varieties, and he sent to us, and we are testing them. And our idea of editing wheat S genes for the blast resistant received the Innovation Idea Prize by the Islamic Development Bank. So it is one of the ongoing project. Our second approach was biological control of wheat blast by using native probiotic bacteria.
Excuse
In
me?
our lab, we have 650 probiotic bacteria we discovered as endophyte of rice and wheat, and we tested 650, and 4 of them showed very good inhibitory activity against the wheat blast. And in the greenhouse, we found a very good protection. When we tested them in the field, we found 70 to 90% protection in the field condition. And we repeated 2 years, and this year also we are testing them. You may ask them what are they? They are basically, bacillus. And we found, we did
Excuse me,
whole
sir.
genome sequence of the bacillus, and we found them. They have the chemical arsenal. Bacillus are very rich in biosynthesis of the secondary metabolites, antibiotics, and they also induce systemic resistance on the host plant. And we have another approach to use nanopesticide, because we have very good collaboration with some nano architect in Australia, and we found that titanium dioxide P25 nanomaterials around 15 nanometer in size can protect the wheat plant from the blast fungus. And this nanoparticle reacting with the oxygen in sunlight can produce superoxide, and superoxide can kill the fungus, but at certain dose does not affect the plant. So this is another ongoing project, and we have a fourth approach, that is we received a good fund from International Atomic Energy Agency in Vienna, and we are using gamma ray, X-ray, ion beam, and now M4 population. We found some of the mutants are almost immune, and this year we are going to do a genomic study for genotyping and understand the molecular mechanism of their blast resistance. I am nearly at the end of my talk. What I feel that in case of wheat blast, we need a global consortium, and not to do a classical research, just discover and publish the paper. Rather, a consortium of global scientists to save the future threat of the wheat blast in the India, China, and other major wheat growing countries. So rapidly and effectively respond to emergencies, we need a new way of doing the research business. And you can find this paper. We shared our experiences. Plant health emergencies demand open science and tackling a serial killer on the run. We can keep our data to publish a paper in Nature, but what we did, we used open data sharing and open science, which has been also practiced in case of COVID-19. All the data in COVID-19, sequence data and papers are, open science and open data sharing. This is why we have now vaccine very quickly. So in case of plant health emergency, we also follow the same procedure. So what should be the take-home message? The Orisam wheat killer fungus, Magnaportia oryzititicum, does not sleep, and it is on the run. And it never asks, any government to get passport. It can pass through the border and infect the new country. So no country is, immune against the wheat blast. We urge global scientific community to join our fight against the wheat blast to find the solution. And we must find a solution before it becomes catastrophic in Asia, because in Asia, food security, would be the greatest challenge, because our population increase is the highest compared to any other continent. So I suggest, and we advocate, open data sharing and open science are fantastic approaches for rapidly tackling any plant health emergencies like wheat blast. I am very much thankful to Professor Arvind Deshmukh, the president of Microbiology Society India, Dr. Dilecta Di Costa, for inviting me and delivering the talk, this webinar. And I am very much thankful to lots of globally, champion researchers who joined with us in our fight against a formidable foe of our major food crop, wheat. And I'm very much thankful to lots of donors who offered us funding, and my team members, my students, they deserve thanks, because all the data they generated under my leadership. So thank you so much, and I am expecting the questions and comments from your side, and I am very much curious and interested to collaborate with Indian researchers on different aspects of microbiology. Thank you so much.
Thank you, sir. First and foremost, let me congratulate you, sir, for that commendable and excellent presentation. Just to summarize of what you have said, and later on we'll start with the question answer session. So it was really nice to know that you have worked for increased food production in just 50 years of independence of Bangladesh. And you were the first, Bangladesh was the first to get this wheat blast, the killer of wheat, which emerged in 2016 due to Magna Ponte Orizae. Now you all were tackling the serial killer, and then you got to making a strip of using the Cas12a smart technology to detect wheat blast at a cheaper rate worldwide. And so much of your expertise, you have not left it only in your country, but you wanted to spread it, and to the scientific community all over the world, by sharing things, we can get more information, and better research can be furnished. And now you have gone to do the nanoparticle technology, which is also a very promising solution to wheat blast. And rightfully you have said that this wheat killer, or the serial killer, does not sleep, but it is on the run. So very rightfully said, and we thank you so much for sharing all your experiences and your expertise with us, and all the technologies that you have used in fighting this serial killer, that is of wheat blast. Thank you, sir. And now I would, I would entertain, or this webinar, we would entertain questions and questions from the audience. So let me just give you a set of rules. So the questions, you all can, in the chat box, you all can write your questions, especially the students. I would request them to write your questions in the chat box. And the others, you all can just, tell me when you all want to ask a question, and I will allow you, so that everything goes smoothly. So please cooperate, and let us get maximum from Dr. Tofazzal, who is ever willing to share his experience and expertise with us. Yeah, so I think we should start now. So can we have the first question from one of the audience members? Dr. Deshmukh?
Yes.
Yes.
Very excellent talk and full coverage given by Dr. Tofazzal. I really congratulate him and
Thank you,
what
sir.
he has expected in his speech. Collaboration in the future. So really we will collaborate together. Our many scientists will make an appeal, and we'll see the collaboration in future with a young and dynamic scientist like Tofazzal. So I would like to put one question.
Yes, sir.
Sir, you have told that you are trying with the titanium as for a nano biopesticides. So again, I am requesting you, there are many other metals which are also very, very effective in addition to the titanium. So anyone in the world is working with the other metals producing nano biopesticides? And can you focus some light on the other metals for the use of nano biopesticides?
Thank you very much. There are lots of nano pesticides, nanoparticles have been used against the microbial pathogens, including plant pathogens, silver or tin or many others. But in our case, we screened lots of antimicrobial nanoparticles, but we found titanium dioxide P25 type, which has been synthesized by one of the leading nano architect, Professor Yusuke Yamauchi in the University of Queensland. It worked very well. When we apply this particle during flowering of wheat, it protects the plant. If you artificially inoculate the plant with high concentration of conidia, even though plant remain unaffected. But obviously, India is very leading position in nanotechnology. I guess lots of antimicrobial nanomaterials are available in the Indian scientists. If they share with us, we have very fantastic, facilities to do, the high throughput bioassay at the seedling stage, as well as in the hotspot field assay. So we can first do at the laboratory in vitro, and then greenhouse and in the field assay. If we find better nanoparticle, that would be fantastic. So I'm expecting as some of the nanotechnologists in India will contribute, because since 2016 wheat blast not infected Bangladesh. All of my laboratory members are now dealing with wheat blast, so it is now invaded to my laboratory. And we developed lots of protocols, and we published several papers, nearly 10 papers in high ranked international journals. So our team is very serious and very eager to collaborate with international collaborators, especially collaborators from India.
Yes, sir. So I think we have a question from Arun Janu. And the question is, what was the timing of the application of titanium oxide?
Yes, the timing I already mentioned. Titanium dioxide, as the disease wheat blast, unlike rice blast, you cannot see a symptom clearly in the vegetative stage of the wheat. You can see the symptom when spike, coming out in the flowering stage, and especially at the neck of the spike. So early stage of the flowering, we spray the nanomaterial, so that if conidia comes through the wind, they can be protected by the nanoparticles. So at the beginning of the flowering stage, we apply the titanium dioxide 15 days interval.
Thank you, sir. We have another question from Sumit Kumar Chaudhary. Sir, can you tell us the symptoms and some major applications, how we would be able to detect and how we can kill the pathogens?
Thank you very much, Sumit Kumar Chaudhary. The symptom is when you see the symptom in the spike, that means in the head, the symptom is like fusarium head blight. But here there is a little bit different. Rachis, you can find blackening in the rachis, but in case of fusarium head blight, pinkish symptom is appeared, and in the older leaves you can find typical eye-shaped lesion. And detection, as we discovered new primers, you can do PCR very easily, quickly you can detect, or our PCRD strip you can easily use. Within 30 minutes you can detect it. And I think the PCR is the definite way to detect the fungus.
Thank you, sir. I think Sumit Kumar Chaudhary, your question has been
Yes,
answered.
yes, ma'am. Thank you, ma'am. And thank you, sir.
Now we have Vantana Murugavel. What are the quarantine measures taken for wheat blast?
Yes, quarantine measure. You see, worldwide there is quarantine measure to check the whether wheat during export import. Wheat is number one export import crop. In that case, there were no diagnostic tool in the quarantine people. This is why it escaped the quarantine and came to Bangladesh and also landed on Zambia. So our PCR strip or the PCR method, very specific, and it could be used now by the quarantine and to stop the pathogen spread through the even seed or grain.
Yes, sir. Thank you, sir.
Yes, sir. Sir,
Yes.
I would like to ask one more question to you,
Yeah, sure.
but not exactly of your research topic regarding microbiology.
Yes. Welcome.
Sir, India and Bangladesh are neighboring countries. We can travel by bus, we can travel by train, we can travel by air. So, last year, few Bangladesh students came to India, had a training in Indian colleges and universities. In India, we have arranged for their food, for their accommodation, and for their training. So, such type of vice versa, if it is possible in Bangladesh. And second important thing, if our Indian student want to do PhD, let us take under you. What is the fellowship possible? What are the opportunities to go to India and work, go to Bangladesh and work with eminent personalities like you for PhD? Can you guide, please, sir?
Thank you so much. You have two questions. First question, in case of student exchange, India and Bangladesh, I think it is a nice idea. We can exchange our students, and from Bangladesh, most cases they visit India and they receive welcome from your side, and also receive lots of training, because many students they received graduation also from India. But in case of Bangladesh, vice versa is not well established from our side. But from my side, from our Institute of Biotechnology and Genetic Engineering, if your student wishes to visit Bangladesh, we shall be happy to welcome them and arrange their accommodation as well as other logistics.
Very
And
nice, sir.
what
Very
we
nice.
and
Thank you. Thank you.
what we can do, we can try to make some arrangement. SARC Agricultural Information Center. We can approach SARC for making such a stable and sustainable exchange between India and Bangladesh. And it is really great idea, and we can work on it, and surely we can find the avenues. And if anyone wishes to work in my laboratory, obviously I can arrange for the PhD student the scholarship, and obviously a student needs to apply beforehand so that I can arrange the scholarship. Our university has a scholarship which is a modest one, but in my group all the students they are receiving scholarship from the external funding. We are going to have a very big collaboration with Global Institute for Food Security in Canada, and it would be 10 years collaboration, and lots of PhD and postdoc opportunities will be included, and it is already on the run, already been started. So I hope we shall have lots of opportunities. In my university, we have, in my laboratory, we have Illumina sequencing platform, the ultra modern one. So if you want to send the sample, we can sequence and we can start collaboration. You see, in case of wheat blast, we had no funding, no project, but we did the big job. And that allowed me to make huge collaboration worldwide. Wheat blast, even are now connected to me. And it is really amazing. If we seriously wish to happen a collaboration, effective collaboration between India and Bangladesh, from my side, I would be very happy, and I am ready to start immediately.
Very nice, sir. Many vice chancellors are my good friends in India. So definitely we will work on it.
So we have a question from Rabea Ruby. 2 questions from her. Can we use any natural fungicide to protect this blast? And the second question is there any side effect for using titanium dioxide?
Yeah, 1 minute. Rukaya madam is from Kashmir. And I request her, you can ask the question. Rukaya, please.
Not Rukaya, Rabea.
Rukaya, you can ask the question.
She's from
Okay,
Rabea.
sorry.
Okay, Rabea Khatun Ruby.
Yes.
Sir, is there any side effect of using titanium dioxide? Titanium dioxide has been used in cosmetics and even in human use. So it depends on the dose. So in case of wheat blast, we use a ppm level concentration, but we didn't check the side effect on other organism, and it is necessary to do because nanomaterials they have sometimes nanotoxicity. So we have to check whether it has any other side effect or not. I have seen another question from Ruhit Raj. How can this disease be checked rapidly before it causes devastation? Devastation at large extent that is becoming a pandemic. Also, in terms of economy, does it feasible for the common lower middle class farmers to bear the cost? Yes, this is really an interesting question. One of the factor is the weather, and the disease is, it becomes devastating at the flowering stage of wheat. It is predominantly head disease. So during the head, when you see the symptom, then there is nothing to do. So there would be the best solution would be if we have the resistant variety, and we suggest our farmers if weather depending on the weather forecast, if there is a chance of rain or heavy dew fall during the flowering stage or heading stage or grain filling stage, we ask them to apply strobilurine or azoxystrobin type fungicide to stop the devastating infestation. Economically, yes, farmers use the fungicide, and other thing is that seed treatment did not work in our case, we found, because there are lots of alternate hosts. So fungicide as well as resistant variety would be the best option. I found another question from Sumit Kumar Chaudhuri. I have another question that there are any easy way to kill the pathogen and which are not harmful for us. Yes, the best way would be if we have a resistant variety. Then no need to kill the pathogen. You can control the disease. Otherwise, fungicide application always not good for the environment. Anusri Ghosh. I am a researcher working in Bangladesh. I have a question. Please ask question.
Thank you, sir. You have a great job and great research that I am feeling. And 2 years ago, I was a student of Haji Mohammad Dani Science and Technology University, and now I am working with the development sectors. And I have a feeling to share you that we are doing a number of researches worldwide, obviously, but there is a lack of communication and collaboration between the government sector, NGO, research organization, and the academia. I am feeling that you are a great researcher and have a great long experience in the research sector. So, is there any suggestion to disseminate the research findings and to fill up the gap so that all can contribute and accelerate the researchers?
This is a fantastic idea. In case of COVID-19, it first started in China, Wuhan city, in December 2019. After COVID-19, what scientists did, they sequenced the virus and they claimed that it is a novel coronavirus, and they released the data publicly. And in our case also, in case of wheat blast, what we did, we use open wheat blast platform. And in case of coronavirus, due to the sequencing, RT-PCR protocol developed, which is a gold standard and have been used worldwide. Very fast it spread like the COVID-19. Similarly, different vaccine designed based on the sequence data, because thousands of sequence data, are publicly available. So in case of any plant health emergencies or any scientific issue associated with food security or human health, what we can do, we can change our classical ways of dealing the science. We can use the open science and open data sharing approaches. We can engage the global scientific community on that topic, and then address it jointly. And obviously, that would allow us to engage the policymakers, extension workers, and if you do everything openly, open science and open data sharing approaches. So from my experience on wheat blast, what I realized that the open data sharing and open science approaches can shape or can enhance the ways of impacting science in the real life. Your question is very important, and I expect the donors, I am advocating worldwide to ask the donors so that all researchers can do the same practice, not just publishing a paper in Science or Nature, rather solving a problem very quickly by sharing the data and exchanging the views and collaborating with the global scientific skills and integrating the science as well as the funding. Funding also, even we can do crowdfunding for handling the any emergencies. Very nice.
Archana madam from Delhi.
Thank you, sir.
Want to ask one question.
Yes,
Archana
please.
ma'am.
Good evening, everybody. I could join late because even my unit was having a function before this. So it was a very, very informative talk by Dr. Islam. But I heard from the midpoint. So I would only like to request Dr. Patankar, Dr. DeCosta, the entire team to please quickly upload it on the YouTube, because I want to hear it full. So I would like to know the strategies told by sir.
Yeah. Okay. Sure.
Thank you, Dr. Archana. All of my lectures, and not only this lecture, my classroom lectures, I always share through the YouTube. I have a YouTube channel. So
Great.
I believe that everything should be open.
Yes.
We should not limit ourselves in the small classroom or a small group. Scientific knowledge should be shared. Even any technology should be shared, and this is why I do. My team always publish paper in Open Access Journal. Our paper was requested to publish as a brief communication in Nature, but they are not open for the developing country. This is why we decided to publish in BMC Biology, and it was cited 200 times within 3 years.
Great.
So it impacts hugely.
Yeah. And also you're trying to share it and benefit the society more and more. That is the real venture of a true scientist. It should be open and it should be available to all.
Yes.
It's very appreciable, and I will look for your lectures tonight itself.
Thank you so much. And if you need any materials related to our research, are free. Any Indian, if want to do research and if wishes to get any materials, even the primers, we can send freely. And you can send your even any microorganism. We can do sequencing and we can do collaboration.
So nice. It's really awesome. And I once again congratulate Dr. Islam sir and as well as the organizing team, and of course fundamentally Professor Deshmukh sir, who made it possible through this platform. Thank you.
So we have another question from Al Amin Shitol. Any specific anti-pathogens that can be used to destroy this fungus?
We used lots of, antagonists, and we found some isolates of Bacillus subtilis or Bacillus amyloliquefaciens. We isolated from the as endophyte of wheat seeds. They worked very well in the field, and we are going to make a formulation for the farmers, and it would be a good biocontrol agent. So in case of Bacillus, they are very famous in producing antimicrobial substances like secondary metabolites, enzymes, and as well as some secondary metabolites that induce systemic resistance in the host plant. But obviously, there might be some other microorganisms we have not yet been tested. If you have good antimicrobial or antagonistic microorganism, you can share with us. We can test it freely in our laboratory and share with you the data.
Yes, sir. Thank you. Sir, we have Dr. Mohammad Arif Uzman, who would like to ask a question.
Yeah. Thank you very much. Good evening to everyone. I know personally Professor Dr. Tofazzal Islam sir. He is a leading scientist of Bangladesh. So I first congratulate him for giving a nice delivery regarding blast disease. So I have a question to him. We know that creating resistant variety is a durable technique. And what about the resistant breeding update in your lab? Because we have seen that you got some resistant lines from Japanese professor Yoko Tosha. I personally know him. He's from Kobe University. So, sir, please tell me some words about your resistant breeding approach.
Thank you so much, Dr. Muhammad Arifuzzaman. As time was limited, this is why I made my story short. Actually, RMG8 GR119, the gene is immune to wheat blast, and the Albanian genotype I received from the Professor Yukio Tosha. I visited his lab. His lab is #1 lab in the world for wheat blast, because they started work in 1985 and till today they are continuing wheat blast research, and they published paper in Nature and Science also. So what we are doing, they gave us the marker. So RMG8 and RMG GR119, two genes we are integrating to our 2NS translocation bari GOM33, which is 70% resistant. So we are making the pyramiding of the gene, marker assisted breeding. And one of the students from Dr. Arifuzzaman's University, Haji Danesh Science and Technology University, Mr. Mujahid, he is working on it, and we received 350 F1 seeds, and this year we are growing the seedling and see whether gene is transferred, and we hope that we will be successful as we have the marker. This is also one of the way, and if you collaborate with us, especially in double haploid technology, because Dr. Arifuzzaman, I heard you are very expert in double haploid breeding technology, then we can get homozygous plant very quickly and we can accelerate this approach. So we have various approaches to fight against the wheat blast and get some good solution before it hit the India or China and other major wheat growing countries.
Thank you,
Yes.
sir. No problem. I will
Now,
cooperate with you. No
yes.
problem, sir.
Thank you.
Now we will take last question from Riddhi ma'am. Riddhi. Riddhi, you can ask. Riddhi Sinha is asking.
Riddhi
Electrical.
Sinha asked a question: is killing pathogens from wheat by any way affects wheat production in quantity, and is this wheat different from natural and normal wheat benefits or merits for our environment? Killing of the pathogen from wheat by fungicide. Fungicide in case of wheat blast is an unreliable technique. In our case, we did lots of field work, because when symptoms is appeared and fungus block the vascular system of the head, it dries up and becomes the white and seedless. So it is not so helpful. But if we can use the biocontrol agent, that would be nice. And obviously, breeding resistant variety by using gene editing or marker assisted breeding, that would not affect the quality of the wheat. But we need to go long way to make a conclusion on your question.
So, okay, we can take Richita's question.
Richita Islam asked a question. Dear sir, your research project Wheat Blast is really appreciating. How much it would be helpful if Wheat Blast resistance outcome applied in Rice Blast? Yes, Rice Blast and Wheat Blast actually same fungus, Magnaporte oryzae, but they are different at pathotype level. At genetic level, they are slightly different. This the host specificity, the molecular basis of host specificity has not yet been clarified. So wheat blast never infect rice, rice blast never infect wheat. And rice blast, there are lots of genes have been discovered against the rice blast. But blast fungus very clever, and they can mutate, and still it is a big fight against the fungus. So rice blast gene didn't work against the wheat blast. We need to find the specific gene resistant against the wheat blast.
I think, yeah, that would be the last question. And there's one question for me from a master student, and that is, the question is whether any research was going on in India. In fact, there was, this wheat blast was detected in West Bengal, and that's the time Dr. Tofazzal was called upon to solve the problem. Sir, can you throw light on that, please?
Yes, in India, Bidhan Chandra Agriculture University invited me to deliver a talk on wheat blast. And actually, in West Bengal and our wheat blast infected area very close. So I went there. I had a nice discussion, but that day-long program was suddenly postponed. And then I visited Shantiniketan to see the great poet Rabindranath Tagore. But we have, collaboration with Indian researchers. When we organize any workshop or seminar, they participate. But wheat blast is a dangerous disease. This is why this pathogen import-export is really tough, and it is a biosafety level 4 organism. But what we can do for you, if you have any materials, even genotypes you think strong would be strong enough against the wheat blast, or other materials, biocontrol agent or nanomaterials, we shall be very happy to do the research and allow you researcher to do work in our laboratory. We can host that, and that would be the good beginning. And we can make a collaborative, bilateral project also. And we can ask funding from the SARC or intergovernmental funding, because this disease has high risk to enter into India, and India is producing 100 million tons of wheat, and it is the second highest wheat producing country. This is why I advocate as a researcher, India should take wheat blast very seriously. And we are ready to support whatever the experience and we have now.
Yes, sir. So thank you so much. I don't think we can take more questions. So whoever has any questions to ask and clarify can directly contact Dr. Tofazzal, and I'm sure he will be ever ready to clarify your doubts. So indeed, sir, India has to be really careful, because you can just see in the last year how the coronavirus came and just took us by surprise. So, you can never doubt the potency or the whatever of the microorganisms, especially fungi, because they're very difficult to deal with and to destroy. So we have to be, really careful. So with this, now I'm going to propose the vote of thanks. An ignited mind is the most powerful weapon on the earth, above the earth and under the earth. On behalf of Microbiology Society India, I would like to express my profound thanks to Dr. Tofazzal for sharing with us his expertise and opinions today and igniting our minds. We are indebted to you, sir, for taking time out of your busy schedule and delivering this excellent talk on wheat blast. My sincere thanks to Dr. Arvind Deshmukh for giving me this opportunity to anchor today's webinar. I would also like to thank my colleague Dr. Aureen Gomes for her help in conducting this session. A big thank you to all the participants for attending the talk and actively interacting with the speaker. I'm sure the valuable inputs and insights of the speaker with regard to wheat blast is enough food for thought. I wish one and all. Before that, I would like to say is I'm really impressed that Dr. Tofazzal has said that whatever research is going on should be shared. In fact, there's this very famous adage saying that too many cooks spoil the broth. But I must say here, many more scientists will superblast the blast. And therefore, we need many scientists and need collaborative research. That is the need of the hour. So I wish all of you a good night, and please stay safe and blessed. This is Dr. Dilecta Di Costa, your host for today, signing out. Thank you. Thank you, sir, once again.
Thank you, sir. Thank you again. And please keep in mind, this is not the end. This is the beginning. 1 hour with you is not enough. So, definitely after COVID situation, I want to call you in India. And next year, definitely you will be in India for one or other conference. Thank you, sir. Thank you. Thank you again.
Thank you so much for inviting me and offering this wonderful evening, which would be memorable for me. And I also feel that it is a good beginning to connect with Indian microbiologists. And I have, profound respect to Professor Arvind Deshmukh and Dr. Di Costa, Dr. Archana, and all others who are listening and who will listen this talk. And let us work together and let us practice open science and open data sharing to face any new challenges in agriculture, new challenges in food security, new challenges in our life. Working together is the best way, utilizing the global scientific community to address even a small problem. We can move very fast. Thank you so much. Wish you safe health and safe and sound health in this pandemic. And I welcome all of you to visit my new institute, Institute of Biotechnology and Genetic Engineering. And whenever you visit Bangladesh, please knock me. And I am a netizen. You can find me in internet whenever you browse using any browser. Thank you so much.
Thank you.
Thank you. Thank you, sir. Thank you.
Thank you.
We'll meet again. Thank you.
Thanks a lot.
Yeah.
Thank
Thanks
you.
to one and all for attending.
Thank you.